All right. Hello, everyone. Welcome to another Build a Cell seminar. I'm going to hand it over to Manu to introduce himself and get the ball rolling today. Thank you, Manu, for agreeing to give a Build a Cell talk.
Hi, everyone. I hope you can hear me. This has been a really fun seminar series I've followed through just for the breadth of the sense of community here. Uh, Cells and living matter is so broad that many points of view can actually fit in the same framework, which is really fun. So my name is Manu. I'm a faculty at Stanford in the Department of Bioengineering and also courtesy appointments in biology and oceanography. I won't talk too much about the ocean today, although there are lots of lessons to be learned because life immersed in the ocean. Um, I think one thing that I was going to try doing today is give a kind of a broad spectrum of several themes, primarily focused around self-learning materials and how to think about cells as self-learning materials, but also how to build some of them. Some of it will be abstract and mathematical, and some of it is quite experimental. Uh, Before I start, I want to tell you all how I started my academic career. I started my academic career thinking about this photograph. I was a computer science undergrad, interested in physics of computation. And in 1981, a famous meeting happened, which was primarily focused on physics of computation, where people were starting to ask the physicality of information processing. What are the limits? This is the meeting that gave us the Ralph Lauder's famous theorem of cost of erasing memory. This is also where the Toffoli gates and reversible computing was discussed and discovered, and many sets of ideas around quantum computing were set in that phase. And it's ironic because always from the history of computer science, it's been known that physical limits and implementation in the context of physical science are the drivers for thinking about information processing. But in certain sense, with the success of computer science also came abstractions where a few sets of architectures got locked in, and often enough, much of the rich history of computer science got hidden to many physical scientists per se. So this was the background. I was starting my PhD degree at MIT, and I started to work on bubbles. And I literally mean bubbles. I built the first computers out of air bubbles. One of the things that I was excited about at that time is thinking in terms of what if information processing could do both information and materials processing, enabling the idea that an operation of a logic gates could actually give rise to true material synthesis, reorganization of physical information. What you just saw on the left is a high-speed video of a flip-flop, one bit of memory. And one of the things that inspired me from this work is starting to deeply think about computation and embodied computation in many different materials, including living and non-living. We've continued that work for many years. Here's a little puzzle to warm you all up. I can pause this video for a second, and you can look at it for a second and stare at it long enough, doodle on your piece of paper to see what would happen as these little bits of physical matter propagate through a very simple ladder network. And since we don't have that much time, I can just show you. This is a synchronization gate. While information is propagating, these bits are talking to each other, and through this non-local interaction, they're able to synchronize themselves so they can arrive at an arbitrary time, but they always leave together. Now, for the aficionados of microfluidics, there is a very deep puzzle hidden here, which is everybody tells you that low Reynolds number hydrodynamics is reversible. And that implies that when you play the movie backwards, this should not happen. Uh, there is a certain kind of symmetry breaking here, and you're seeing the thing that came in now synchronize or desynchronizing. So there's a little trick here, which if you think hard about it, you can crack it. A fun puzzle. So anyway, this is how I spent my PhD. When I started my lab, one of the big things we did is we started scaling these platforms. We do a lot of instrument development. I'm not going to have time to talk about it, but just needless to say, we built a synchronized version of these sets of systems to really explore uh, fairly complex systems. At this point, we can build arbitrary droplet processors that can do fairly complex operations. Um, but we turned our eyes to thinking about not just visceral and very direct implementation of computation in physical materials, but much more subtle ways of thinking about it. And I think we turned our eyes to chemotaxis. For many years, we worked on chemotaxis and asking ourselves a simple question: what is a minimal system that can demonstrate chemotaxis? I know many people in this community are interested in artificial cell and artificial cell behavior. And for any of us to be thinking about Darwinian evolution in many of these artificial systems, we have to fundamentally implement complex behavior for these sets of systems to evolve. Now, this is a behavior that most of you have seen. This is a white blood cell chasing around a bacteria. If you ask yourself a question in terms of activity and sensing being the two key parts of this puzzle, how would you go about implementing it? And after a lot of thinking and some serendipity, we were able to actually discover that chemotaxis is a property of every binary fluid. Literally, abiotic systems. This is an example. I kind of like this in the framework of thinking about implementing complex interactions in fluid droplets. So what you're looking here are just food coloring, quite literally. But what's important here is these are binary fluids, where there are two fluids in this droplet which have a different surface tension and vapor pressure. The motor in this system is Marangoni flows. You can actually see this toroidal flow. And what is very subtle, which arises in sensing, you can watch these droplets can sense each other, they can talk to each other, they can push each other. That comes from vapor fields that they generate. They have a sense of self. And again, this is another puzzle you can think a little bit about. How would you go about implementing something like this? So this work taught us many years ago that it's actually fairly simple for complex behavior to arise in sets of systems. And now we have worked on ternary droplets, a much higher order complexity in these sets of systems itself. They also have a sense of self. You can do self-reflection, which is quite a puzzling scenario that the droplet can see itself in a mirror. And one of the things we've been starting to do is to really explore what happens when large number of these sets of systems actually interact with each other. And finally, another physical system that I just want to bring to people's attention, there is some amount of work we have done, is literally foam itself. Foam has been studied in the past in context of tissue mechanics. We've been asking ourselves a question of how to build information processing systems using foam. This paper is online on soft matter. And we figured out a technique to make foam active by programming any voxel in a foam to become active by implying breathing. We're able to do information propagation over long distances. So what you're looking on the left is a stroboscopic video of what we call active foam. Traditionally, you all know foam is a very frustrated material. It would remain stuck in a certain state. But now we can inject energy in the system, and that leads to this incredible, very long distance communication. So on the right, what you're seeing are what are called topological transitions in the system. As we inject energy in the system and stroboscopically bring the state of the system back, topologically, the system evolves as a function of time. And using this, we can write information in this physical system. We can read that information back using local rules. And one of the most fun things that we have recently discovered is a hidden chiral modes in foam, which allow us to really think hard about how some of these simplest materials that we have thought to be quite dumb or just stuck can actually be used in remarkably new ways. So this is kind of a background and history of how I started my career in physical sciences. But of course, as all of us have this fascination, when you see a living system, kind of much of this gets escalated to orders of magnitude. And I started spending more and more time in the environment, just observing, rather than a tendency or a desire to wanting to build and control. I learned this fairly quickly that it's actually very important to also spend time observing. And so the stories that I'll tell you about today in a little more depth are really around observations, and that has led us to think about how cells incorporate behavior and algorithms, and also how we can harness that to build systems that can do self-learning. So there is a zoo of animals that we work on, single cells, larger animals. But asking a very simple question is, how do systems that don't have a nervous system implement complex behavior? So to tease my neuroscience friend, I often call this neuroscience without neurons. Now, we do this as in the context of tree of life, all across the tree of life. And I think it's very important to remember if you're looking for motivation, you should look very broadly in the tree of life to really try to understand and think the beauty and complexity of what's been implemented in the past. So let me just take you through a zoo. This is just to motivate many of you. Just remarkable geometry exists in living cells. And we started asking ourselves this very simple question. How does geometry encode behavior? Could geometry be one of the methods or methodology of implementing algorithms in behavior, which would give rise to real-time behavior of some of these cells? So I'll give you one example. And I think, just cellular morphologies are just remarkable and everywhere. And I think just to give you a historical perspective on this field of learning in cells, sometimes there are directions or diversions that happen. So there's a very famous book by Jennings in 1906 where he described behavior of lower organisms. At that time, he was criticized dramatically because everybody wanted to put humans at a pedestal high up. And what does it even mean for a single cell to have behavior? And many of his experiments were actually said to be flawed or kind of fake. And one of the problems with some of that is that although now several people have actually repeated his experiments to demonstrate learning in Stentor and many other sets of systems. And the ironic part of all of this is uh, some of the work that came afterwards really took this idea of learning in cells quite literally. And this truly was a mistake. When cytoskeleton was being discovered in single cells, several people started thinking about a one-to-one map with the nervous system and called this the proto-nervous system of a cell. Now, this was absolute baloney. Uh, this is a massive mistake in the field. People started labeling many cytoskeleton elements as literally one-to-one parts of a brain, looking for an architecture. 50 years passed until beautiful work by several Japanese groups showed that this is not how behavior in a single cell works. There is absolutely no one-to-one map or thinking about this is not an equivalent of a nervous system as people would have thought about it. But, I think it's a little bit ironic that set this field many, many decades behind. And what is ironic, what I'm about to tell you is actually cytoskeleton can embed incredible behavior and can embed learning rules. So we're reviving this, but in a different light. But it's important to understand mistakes that were made before. Okay, so let me actually just tell you quick sets of stories. And I think all of the behavior that I'll talk a little bit about is written in this framework of what I call perceptive machines. The idea is that there is a world, there is an internal state in a system that we care about, and the world has sensory inputs, and the internal system makes actions and internally also changes its state, and the system evolves as a function of time. And we've been thinking about this in an abstract manner to build an entire hierarchy of machines. These are some of the examples of the machines. This is a story that we're just writing up. that allow us to even understand complexity in a new light. Because effectively, when you start thinking about behavior and engineering behavior, we need better classification schemes that are not driven by sets of desires that we might have, but driven by the functionality associated with these sets of behaviors. So I think I'm quite excited about this abstract framework. And this abstract framework will be out soon. And I'm curious actually if it will apply to many of the systems that people in this community are building. Okay, so let me tell you a few sets of stories. And I know I'm already way slower. So I'm sorry, I'm going to go exponentially faster as I keep going along. But let me just tell you the first story of a cell encoding behavior in geometry. So the cell that we've been working on is called Lacrimaria oler. I discovered it in a swamp with one of my students. We'd been searching for this for almost a decade. So there is value of just pulling up your sleeves and going to swamps but this is what we saw. And actually we discovered this with a foldscope. The $1 origami microscope that we invented a long time ago. So anyway, here is one example of a cell. Just remind you, this is a single cell. It's not an animal. It's not a worm. It's a single cell. And the video that you're watching is real time. This is a mimicry of what we saw under the foldscope. And right there, you can see the organism is going to essentially, it's a predator. It's trying to attack. Now, the big question to ask is, where does all this behavior come from? You see, it's almost real time, and it has the capacity to strike, completely transform itself, and essentially eat this. One of the things about this is we brought this system to the lab. We were able to culture it, which is extremely painful. But over many years, we've now been able to maintain these sets of cultures. You can notice they're not cannibalistic as far as we know, but they are searching for food, enabling just this very complex behavior. So I'm just going to show you another video of what that actually looks like. So you can see the cells, and you notice this neck-like structure. That's really where much of the behavior is. Majority of the time, the cell sits in a place and searches. And the question is, where is the algorithm for this search? Also, there is a little bit of a puzzle here. It seems to be extending this neck as if it's coming out of nowhere. And I'll tell you about cytoskeleton associated with that in a second. But let's just watch that up close. So first of all, here is that cell again. And you would notice very quickly that on the surface, it's covered with cilia. And just from an anatomical point of view, we're going to call this pearl-like structure the body, a thin protrusion the neck, and the tiny tip the head. And again, this is anthropomorphizing a cell. It has no notion of body, neck, or head, but it just makes the language a little bit easier. And let's just watch that strike one more time. You can see this was the poor cell that now is being engulfed in real time. And what much of is known about, there are toxins that are released at the moment of contact. But one of the first questions we were interested in is, where is this behavior encoded? Where in the cell would you encode this real-time search behavior? Now, we build a lot of tools, and this is a platform that's online. Many of you can learn about this. We build a lot of open source microscopes called squids. And this is a modular system. So we built these to be able to do long-term tracks. And we can map the behavior of billions of these cells just over time, searching for food in different types of environments. And that we showed in the first paper in Current Biology, led by a postdoc, Scott Coyle, who now has his own lab, that this animal or this single cell has a beautiful search behavior. There are no blind spots. And what's ironic is it does this in a timed manner, that there is a dormant phase, then there is active phase, and then there is a dormant phase. And you can actually see when I show this as a point cloud, so what you look on the left is every single dot has been where the head has been for a few seconds. And you notice that there are no blind spots. And in fact, we demonstrate that this is actually a random point cloud. So somehow the cell has figured out how to randomize its location. Now, there is also a beautiful scaling law associated with this as a function of the length of this little protrusion. And much of this can be encoded in the buckling modes of the cell itself, the beam that it's using. And in the next paper, we were intrigued, where would these buckling modes be encoded? So we decided to take a very computational approach to this. So just to get you accustomed, we are now building this system in physical. So it's important to understand what's happening here is ciliary reversal. So you can see the surface of the cell is completely covered with cilia. And every time the cell, there is a calcium activation pulse that leads to reversal of cilia, and that leads to retraction, while the head cilia will turn on and off. So you can think about essentially now the head cilia is on again, and you can see that the protrusion is moving forward. And at a moment right there, you'll see suddenly the head cilia will shut down right there, and then that leads to an extraction. So there's a point force at the tip that's leading to on, off, on, off, on, off behavior. So I think the magic is that you have this program, which is just literally ciliary activity. We actually believe that this is completely random. That leads to a mechanical controller that then has to lead to behavior that has to be beneficial for the cell, which is a random search. Now, we can write this in an elastohydrodynamic framework where you have a thin elastic filament with a force attached on the tip. And something quite remarkable falls out of this. This is the work done by Deepak Krishnamurthy when he was a graduate student in the lab, and it's published in PNAS a year ago. And one of the beautiful things is that we can now balance activity and stiffness of this filament to ask what would a filament like this do as a behavior. And since I don't have that much time, I'm just going to show you a video. This is a complete simulation of essentially activity turning on and off. And you notice immediately buckling modes, but you also notice that the object, the head, is actually going in different directions. Now, what's very important is that this is not the classical Euler buckling. Classically, if you take an Euler beam and you were to compress and extend it, it would just show the same sets of patterns. But something quite remarkable happens here because this is a non-conservative force, or what is called a follower force. So I'm going to demonstrate this in a very simple way. So first of all, let's just watch what happens when you run these simulations. It's quite fun to see. First of all, in a certain regime of activity and stiffness, these are periodic oscillators. So just watch the one on the right. If you notice, this is a 10-period oscillator. And although you don't notice a pattern, you will actually see pretty quickly that it goes to the same location every single time in a code. 1, 2, 3, 4, 5, 6, 7. Now, this encoding is actually just encoded in this geometry and elasticity. Now, most of the time, there is enough noise in this elasticity, so this is actually a chaotic oscillator. And what you get is by just putting in any input and output, you get this complete coverage of space. And we were able to demonstrate that this method is completely capable of encoding space, just like what happens in a single cell. So now this is, in my mind, a very clear example. I think over many years, people have postulated chaos playing a big role in cell biology. This is a very clear example of chaos theory enabling cell behavior. And this is not that dissimilar if you were to water your garden and hold a garden hose and let the water run wild, and you'll notice that it'll immediately become a chaotic oscillator. And we were able to actually biologically also show that there is a one-to-one map between the behavior that's seen in biological cells and what we can show in this very, very simple model. Now, what's quite beautiful in the context of build a cell analogy, as we are starting to now build this physically, we realize that there are ways to actually program this, either using amplitude modulation or frequency modulation, or mixing these sets of low and high frequencies can be used for homing behavior. So there is a lot more that I'm not talking about, but this kind of behavior, it's a chaotic oscillator that does have certain control parameters and can actually be controlled. So as a good example for building out of a system like this. And this is the experimental results and the simulation results showing exactly the same locations where the cell also has low frequency or probability of reaching out, and the similar scenario being shown in complete simulations itself. Okay, I think I told you a little bit about encoding of behavior, but I mentioned this puzzle that bothered me for almost 6 or 7 years now of where does the material from the neck come from? So I'm going to play this video. Uh, this is a video from my friend who's in Japan at that time. This is that same cell. And when we get wild type cells, we find this very regularly that the cell is able to generate in real time millimeter scale appendages. So now I mentioned to you, this is a 40 micron cell that is generating 1 millimeter long neck in real time. And so if laws of physics were true, this doesn't make any sense. Where would this material come from? Where would membrane in a single cell? You can't synthesize membrane that fast. You can't synthesize cytoskeleton that fast. And every single time you will notice that not only can it extend the membrane, in a fraction of a second, it's going to spool it in. And quite literally, it's actually spooling it in right there. That thing just disappeared. So we started thinking about this. And long story short, this happens to be the largest example of extension rate and strain in any given cell. In terms of, you can think about there are lots of very long cells like our neurons in our body, but this is being made in real time. You cannot do this via synthesis. And, we can do these torture experiments. So I don't know if this cell feels pain. I don't think it would feel any pain for this. What we're doing is we're holding the cell. This is work by Ellie. She just graduated from the lab. And this paper just came out, and we were able to show that this is an extension that somehow occurs. And now I'm going to tell you where this actual material comes from. When we looked at the cytoskeleton, there came a big surprise that there is an internal cytoskeleton, which is this beautiful hierarchical cytoskeleton. This is a helical architecture. And now it might appear in these sets of pictures that this is a basket-like architecture like Chinese fingers. It is not. What this picture is just a Z projection. It truly is a helix with a variable pitch. And now what's happening is that this helix is microtubules. They're anchored to the surface of the membrane itself. And you can see in the folded and the unfolded state, somehow the microtubule has to arrive from somewhere. And we started spending a lot of time deeply thinking about what does it mean to build cortical architecture. So if any of you have not seen cortical architecture of ciliates, there's some of the most remarkable architecture that has been built. And one of the things we were able to quantify in this is to first demonstrate that these architectures are not single microtubules, but bands of microtubules that align along the grooves in these membranes. These membranes we discovered have pleats, and the microtubule architecture is actually multi-layered. So what appears on the surface is only the surface side of the story, but there is a contiguous architecture that wraps around multiple times. And one of the beautiful threads on this is confocal data. I'm now cutting through, and you can actually see very clearly multiple rings. And so these helices are truly spools. They go on both on the surface, but also internally to store a much larger amount of microtubules. Now, one of the beautiful things about this is that the same goes for the membrane, and the membrane is pleated. And I'm just going to jump to show you now the TEM data sets where you can actually see that the membrane in the extended neck is smooth, while in the main body is actually pleated. And what came as a big surprise was the fact that the pleats are programmed by these microtubule ribbons. Now, why would you make such a strange architecture? When James Webb Telescope was sent to space, we were all worried about it opening just once. And what we discovered in this process is actually this is a single cell that exploits a trick from origami. I was in Japan visiting some origami artists, and it dawned on me at that time that, wait a second, this could actually be origami. And with Ellie, we were able to then prove that the cell actually opens and closes and stores membranes and microtubules in an origami. So I'm just going to show you actually how this origami works. So you can watch this thing video on the right. What it is a pleated helical architecture. And I'm just going to open and close this. I've painted the blue for you to see the hidden portion of the pleats. Now, what's quite beautiful about this, and why I thought I'll mention this to this group, is that the cell is using a topological singularity of D cones moving along these helical paths as a way to control this origami. So tens of thousands of times the cell will deploy this architecture, and not even once will it fail, primarily because it's impossible for this system to open and close any other way. So this is a non-affine structure. Unlike a spring that will just extend and contract, here is a system that only opens as these topological singularities travel. And of course, they have dependence on the sets of angles as you think about. And then this is really the one where you can notice that a lot of material can be stored in this. Now, it's kind of often fun to think about what other objects do this. Here is your regular straw. Now, they also have pleats, but you notice that those pleats are not helically programmed, so they can open stochastically. And that is not the case in this system. So unlike this spring that can open anywhere stochastically, for these sets of helical pleats to open, they can only open as these singularities travel back and forth. And this is a quite a technical point, but actually it's not just a D-cone singularity, but also a twist singularity in the microtubule that enables this architecture, because the pleats are programmed by a ribbon of microtubules, and this ribbon, as the pleat open, has to flip up. And so there is a 180-degree twist. And in the physics literature in carbon nanotubes, these are called twistons. And there are 14 twistons traveling on the neck as these things extend and contract. So it's a really remarkable control system that's built primarily using geometry. This is really the big kind of a message that I wanted to share, that you can embed controllers of cellular behavior in geometry. So these are the 14 twistons that are now all along, and they also interact with each other to be able to make sure that this neck will do what it needs to do. And, it's kind of interesting. We've been able to build these sets of architectures, but very, very rarely we can find cells that will have a pleat defect. So this is an example of a cell that biochemically is completely fine, but it actually has a pleat defect because the D-cones opened at the wrong location and it cannot come out of it. So, just to give you a sense of for why geometry is important. So I think I'm already running out of time. I was going to tell you a little bit about how algorithms encode mineralization, but I don't think I have that much time. So let me just jump and end with, because I do want to have time for questions. Let's talk briefly about artificial self-learning materials. One of the things that, so this is going to be a little bit of a switch, but I want to take geometry to another level. In terms of asking ourselves a question, are we just stuck by thinking about sets of learning by only looking at what we can find? And as we started realizing this, the fact that cytoskeleton could be doing remarkable number of things that we just as yet don't have tools to dynamically observe. So we took a step back and we asked ourselves a very simple question, that purely in geometry and mechanics, what could we implement? And then the challenge we put on ourselves was, could we implement unsupervised learning in mechanical networks? And now that's, I wouldn't be telling you this if we weren't able to do this, but now I'm very excited about looking at these sets of ideas and building them with molecular scale. But it was important for us to take this step back. So let me walk you through this set of an idea. And this is work with a postdoc in the lab, Vishal, and a graduate student, Ian. Vishal did much of the numerical part of this, and Ian did much of the experiments. So what is the big idea? I think what we were interested in is, can we build a material that follows the laws of physics, is made out of just purely local interaction like springs, has the capacity to read the environment and adapt and change to learn a specific function. Now, this would be the classic framework of machine learning, except one of the important criteria for us is we really cared about unsupervised learning. I think a shoe is a great example. What if you could wear a shoe that when you walk on a rough terrain, learns that you're actually walking on pebbles and changes the stiffness? We don't have any sponsorship from Nike, but I kind of enjoy the idea that everyday material that we all care about, I wish it could change. Uh, and of course, much of this is really thinking about how could we build and design these in artificial systems. So 3 simple rules. We want it to be self-learning, we want it to learn continuously, and we want it also to be purely mechanical. So no batteries, completely driven. The energy of this learning comes from the environment itself. So I'm just going to tell you the conceptual framework for this. The first thing we thought about is let's build certain class of springs that could change stiffness. Now, that's a fairly easy idea. Your memory foam does that. Lots of materials out there can change stiffness. The second one was this idea of threshold activation, which is that it should be able to take a certain input and beyond a given threshold change. This is almost necessary for us to be implementing neural net-like architectures. But the third rule is a little bit tricky. We wanted it to follow Oja's rule or be a mechanical analog of a neuron. And so we wanted it to be directed. Now, this is a very strange thing to say in mechanics, because you all know reciprocity applies in many of the classical sets of frameworks. So we're saying forces can propagate back and forth through the spring, but information of update rule only occurs when the head and the tail have a slight asymmetry. So this is the directionality associated with this system, that the system will change stiffness if the amplitude on the tail is lower than the amplitude on the head, for example. So, this is graphically, I can represent this. On the left is neurons. Neurons do this directionality through spike timing dependent plasticity. In neuronal dynamics and unsupervised learning, this is implemented through Oja's rule, but we wanted a mechanical spring that could do something like that. And, I wouldn't be telling you this if this was not possible, but I just want to share what does it take to build by design. Now we have these sets of rules. Can we actually design a physical material that could do this? And so the physical realization is right here. I'm going to play this video, and I'm only going to talk for another 5 minutes and then transition to questions. I'm seeing certain sets of questions in chats, but let me just finish this thread and then we will have 15 minutes for questions. So let's start with, firstly, what you're looking at is this little toy. It has a little spring around it right there. And as you notice, when I put input into this material, this blue bar changes and the stiffness of the spring is changing. What this implies is that the force that is transmitted through here changes. You see, it's not jiggling that much and then it starts jiggling more. But now I take the same object, I flip it 180 degrees, and now I make the tail the head and the other way around, and you notice it does not update. It's still transmitting forces, but it's not updating its stiffness. And that's why we had to invent a new symbol for springs, which is it's a simple spring, but now with an arrow. So you will very soon see that we will be able to make topological mechanical circuits, because forces propagate in this network both directions, but the update rule is directional. So first of all, how can we actually do this? So this is a closer look. You're now watching, we are updating, the spring is rotating. This is the direction in which, and it's quite a robust system. It has an internal degree of freedom. This pendulum is how it extracts energy from the environment, and it's used to drive the adaptive part of the system. But I think what's important is, although it's built in a complex sets of parts, this is quite a dynamical system. It's quite robust to perturbations. Now, what's quite exciting, and I think I'm just going to show you this very graphically, although we can demonstrate mathematically where does this directionality come from, but just this is the context. What's hidden behind that spring is a 4-bar mechanism that implements a unique gating function. And that gating function is the drive and the gate for it to pump energy into this rotating spring. And what you notice here on the left, the spring has an asymmetry. And this asymmetry of the directional spring is what determines the change in stiffness. So you can see it's thick on one end and thin on the other end. And this is a very simple way that we actually change stiffness. And for it to be able to update, the gate has to be open and the drive has to be on, and that only comes through directionality. Now, I think I'm going to skip this to just show you certain things of what we can actually do with this. But what's quite beautiful is it's a parametric oscillator. We can write this down explicitly as a dynamical system, and we can then start building networks. So you'll watch two of these networks. The first network is a triangle, and the second network is a diamond. And you can notice that the springs, every color of the spring are updating differently in these sets of systems, and this is encoding what we call a mechanical circuit. Now, what's important is if I had taken this mechanical circuit and just flipped the spring in the other direction, it would actually have a completely unique behavior in encoding for whether it will have steady states, whether it will have oscillators. So I know many of you work on genetic circuits, and you know you can build fairly complex positive and negative feedback loops. Now we can do exactly the same sets of things, but purely using mechanical elements. And what's very interesting is that we can model these sets of systems to demonstrate the kind of behavior. So this is a fun set of circuits, and you can stare at them a little bit to see if you can predict why this system does what it does. So let's just take this as an example. I can see information propagating, and you can see both the blue and the purple become stiff together. Now the green also becomes stiff at a certain point. You notice that the orange, so there is stiffness that's bouncing back and forth. This is a little bit in sync. These two are a little bit in sync. You can see information flow in this loop. You can see there is another information flow. Now, this is an inhibition cycle. So we have certain class of rules, which are design rules that we can use to also design these sets of circuits. And then I think the other thing I wanted to mention is functions. Since I mentioned this in the context of learning, we've been able to show that we can do pattern finding using a system like this. So we can directly learn a given pattern, for example, the roughness of our environment, and let the springs adjust to it. And then again, it's continuous learning. I can give it a completely new environment and let it encode another function associated with this. Now, this example only has 3 springs, but of course, the computational capacity of this thing is driven by the number of springs. So here is one example. On the left, you're watching a completely classical elastic network with these white nodes where we are injecting energy. And you notice that the network doesn't adapt, and so those sets of inputs remain the same. But now look on the right. On the right, we have encoded this system to essentially almost be stealth. So it's trying to hide the places where the energy is being injected by stiffening the network right around that. And this is just one example of literally 10,000 or 100,000 networks that could be implemented because these networks are topological. So every arrow that you change in this can actually change the behavior and what is being learned by these sets of systems. Now, the reason we're excited about this is actually twofold. One is, when you can write down these things as sets of equations, I had thought that we would be able to find an electrical analog of this type of a learning rule, but I haven't been able to find. It's not a memristor, it's not a quantum flux parametron. So I am quite puzzled. And so if anybody has ideas on electrical analog of this sets of idea, because often enough, there should be both analogs of this, and if not, it would actually be fun to invent that. And then secondly, one of the big things that we're quite excited about is to be able to build these class of materials in the context of physical functions. I was joking about shoes. We're building lots of different functions associated with this. But also, we potentially have now the capacity to think about whether cytoskeleton could implement a rule like this. Now, that's completely just a hunch. But before this work, it was not clear what type of computational capacity you could build in elastic network and unsupervised learning approaches. But now I'm quite excited about the fact that we understand a framework to implement these learning rules in these class of materials. And what I'm excited about is both exploring whether physical systems, especially primarily using biomolecules, are capable of implementing this rule. And then secondly, could we actually build and design a molecular analog of this gizmo that I told you about? Now, this gizmo looks quite complicated, but actually it doesn't have to be. There are already many much simpler implementations that we have built. This is another implementation that's on the archive paper now. But overall, we're quite excited about thinking about simplifying this framework itself so that we can build large class of materials that can demonstrate unsupervised learning. So there's lots of other things I wanted to mention. Maybe I'll just mention one single image of this, and then you all can read the paper. Uh, We've been thinking quite a lot about how cells encode materials, and especially the rules of biomineralization. Many of you know biomineralization is just absolutely crazy in what it's able to do. So we've been working, and now this paper is also on archive, where we have shown a really interesting rule utilizing cytoskeleton networks to actually build and design and program biomineralization. And these sets of biomineralization, so this is one example of the system that we work with, that every single shape that you see is actually made by single cells using a programming rule. And we've been able to identify these rules. And kind of the one-liner of this paper, if anybody's interested in reading on bioRxiv, is we've discovered a top-down approach for how cells can program inanimate materials using mineralization. For the last 150 years, people have been looking for bottom-up approaches, which are primarily proteins that are synthesized that can change lattice structures. What we've been able to show that it's not just bottom-up, it's actually bottom-up and top-down. And both of these approaches are critical in building and designing these mineral structures. So I think I'm going to stop here. That was 45 minutes or 47 minutes. Sorry for running a little bit. I'm just super excited about chatting, conversations, and questions. So
It was
fire away.
definitely worth the extra time. That was thrilling to see all your gadgets and gizmos aplenty, of course, and a very engaged audience. We have a handful of um, questions in the chat. We also, I've noticed some people just making friends in the chat, and that is so fun to see. So, for the purposes of the recording, Manu, if you want to read through the chat, and then if you have any questions that you'll be answering, read them out loud for the people on YouTube later who can't see our chat.
Yeah, I mean, people can just, if you are here, just raise your hand and I can just call on. It's much more fun because you might have typed it in. I might have answered that question or not. So it's much more fun. I see Arnie right there, and I don't usually call favors, but I have to call on Arnold. We spent lots of time together. So Arnold, you can go ahead.
Really, I wouldn't want to go ahead. There's lots of questions in the chat. Maybe you want to go through those first. But yes, I do have a question.
No, just go ahead. I'll listen to you while I'm reading the chat.
Oh, okay, cool. Yeah, so my question, Manu, amazing, right? These self-learning networks. And really my question is, how can we build this in a cell? How can you have molecular components that will have similar functions at the micro scale, right? So one thing you
Yeah.
mentioned is that this little pendulum that you made basically is an additional degree of freedom.
Yes. Internal degree of freedom.
Exactly. Yeah. So if you want to make that at the micro scale, let's say a protein or a macromolecule, how would you build that in? Because you can't have inertia anymore. You can't have things
No. I
that
think it's
rely on momentum. So
yeah.
you need
I think
elasticity or
the
something, right?
biggest thing that I'll just mention is And again, this is a provocative comment, and I'm actually actively thinking about how to build these things. But the thing that I want to mention is when we look at proteins and biomolecules, of course, now cryo-EM has allowed us to see just remarkable architecture. But I don't think we as yet still see all the hidden degrees of freedom that are at play. Of course, now with more and more sets of practices of getting all the states, much of what we discovered about how molecular motors move, and people still debate about hidden states in many of these sets of systems and hidden degrees of freedom. So it doesn't have to be inertia, but it's actually, we should be thinking about proteins as much more complex machines, because I feel technologically we've spent an incredible amount of time and effort in getting this atomic scale architecture. But when I read the function, our mechanical understanding of that function in my mind is still quite rudimentary. Simple things like, oh, the gate opens and the gate closes. I can understand. But allostery and many sets of examples have shown that there is remarkable degrees of freedom in these sets of systems. And I think the approach that we are starting to take in something like this is start by building and shrinking these systems. So already one system that Ian is working in the lab, where we have been able to show that we can build this type of an architecture without this inertial framework and also using compliant mechanisms. So we're kind of moving towards compliant mechanisms as another mechanism, which is much cleaner to be able to implement. So I think, long story short, we're not there yet, but I think I am quite convinced that as yet, when we define function to a protein machine, we are ruling out because, of course, it experiences forces much larger in range than what we might see in its sets of states. So I think there is a lot more there, and especially in interfaces of proteins itself. So when you see a much larger macro scale molecular machine, then it begs the question of what does that interface actually do? We don't have that intuition for friction in many of these systems. So I feel from a biophysics perspective, there is a lot to unpack, and the number of structures keep coming out. And of course, one approach would be is to build and design it.
Very nice. Thanks. Thanks, Manu.
Yeah. I see Jan's hand. Go ahead, Jan.
Oh, hello. Hi. I just had a quick question. This garden hose instability that you were talking about, it really looks like a foraging strategy. And I
Yes.
was just like, this might be a very stupid question that you might have touched on the paper, which I did not read. I'm just learning about this now. Like how does this project, the cell origami structure, combine with the second project that you mentioned, these learning materials? I'm assuming it has something to do with like the feedback process of the pose thing, and how these two, because I see some overlap, but I didn't do any deeper insights, and I feel like you have an answer or some
Yeah,
comment.
no, I think it's not a direct answer. I just wanted to share the fact that geometry can actually be used to implement algorithms, and very robust algorithms. So what's actually the key message to take away is that there are two topological singularities that are completely hidden in such a simple architecture like a helix. I mean, we've seen helixes all our lives everywhere in biology, and it took us really 6 years of banging our head to figure out, wait a second, this thing can actually be controlled using a really beautiful mathematical architecture, which is scale-free. So I think the analogy there to think about is, as we are building and designing cells, we have to strip away many of the abstractions that we are used to in the current framework of how we build machines. And geometry plays a deeper role than what we actually realize in cells. And so, when you think about learning now, I can tell you that already, if you take a look at the pleat architecture, we've now compared that cell to many other cells that have similar architecture but are missing this helical architecture. So we are able to then show how novelty in behavior or how evolution of cytoskeleton geometry can actually lead to function. And I'll kind of show this image because it was years of work for Ellie and I. We kept searching for these sets of species. So I just want to share this. Now, here are 4 cells that seemingly look very similar to Lackery.
Mhm.
But then when you look at their behavior, they do nothing like that system. And you can then imagine how novelty arises in behavior. This is much more like an elephant trunk. And then when we look at the cytoskeleton architecture of many of these species, they have all the components except they're missing this twist. But without this twist, you cannot actually implement this. So I think the analogy to think about here a little bit is not a one-to-one map, but just the fact that geometry goes a very long way.
I see. Okay. Thank you so much.
Any other questions? I see a question by Mohammed Sufi. I don't know if you want to read it, Mohammed, or I can read it.
Yeah, I can read it.
Go ahead, Mohammed.
I mean, I wrote it in there, but Johnny just wanted to ask about what people
Yeah,
are
just
referring
ask
to.
your question. It's more fun that way.
Yeah, sure. Yeah, we can do that. I wanted to ask if people try to reconstitute the ciliate neck geometries or other geometries that you've shown. And I wanted to like understand, like, what are you thinking about for the minimal system to recreate this behavior? Because that's something we're all interested in the build
Yeah,
itself.
I think we are working on a reconstitution for that particular system. It's not so trivial. You have microtubules, you have membranes, you have activity that you have to add to those sets of systems. I mean, this is a very recent discovery, so it'll take time. One of the aspects of this is we can reconstitute them at many different scales. So currently, we've also been able to actually show that there is a lot more in that geometry. Ben, who's a postdoc in the lab, Ben and I have been able to actually now implement a general purpose version of that type of an architecture that has a lot more degrees of freedom rather than just one single extension. We can actually program any arbitrary curve in 3D space using that design. So it's quite broad in some sense. The other aspect of it, if there are some reconstitution people here, we're very interested in microtubule ribbons. They are non-trivial to make. Of course, you can make microtubule bundles, but ribbons have certain sets of properties. And so that's another kind of a big task on our hand, working with a couple of labs. We're trying to build sheets of microtubules. That's kind of all I can say on the reconstitution side.
Thank you.
So from the chat, I can pull one from there.
Yeah, go ahead.
Hannah Burdig says, awesome talk, first of all. Could we implement something like directional learning between non-living liposomes communicating via small molecules?
Yeah, I mean, maybe a couple of points. With liposomes, there is an analogy to what I showed in the earlier version of the talking droplets. They do that by emitting a vapor field. Now you can discretize that and think about that in the context of liposomes. The directional learning is quite non-trivial. It's not just if you have a certain set of an identity, possibly with liposomes, that the liposomes emitted by tail versus the head are not reciprocal, they are labeled, then I could imagine that could be detected and you could detect the direction associated with it. And frankly, when you start thinking about synapses now, synapses come to mind where there is also a directionality, and that's really is the directionality where we were inspired by a neuron and the fact that it's such a polarized cell to begin with. I think in terms of communication via small molecules, that is something that we are quite excited about. Because even in that earlier droplet system, now there is another published work building upon this by Nate Lab, where they have shown what are called trifluids, which are three miscible fluids. So now there are three species of vapor and small molecules talking to each other, and you can think about combinatorial codes associated with it. So I could imagine the directionality coming in from the code itself and not mechanics. And if somebody wants to talk about it, I would be very happy. This is something that we're very excited about. Any other questions, comments, ideas?
I do see a question from Lynn in the chat. Oh, I'll let you go ahead.
Yep.
Go ahead, Lynn.
Great. Hi, Manu. I don't think we've met before, but I've been an admirer of yours. I'm over at your neighbor over at NASA Ames, and actually, I was thrilled that you mentioned Herbert Spencer Jennings, as he was my academic great-grandfather. So I have two questions as a psychologist through Tracy Sonneborn and Johnny Preer. Anyway, ciliates, of course, as you alluded to, have all the subcortical alveoli, which gives them a lot more firm structure. And so I noticed there was a question about putting this system in a liposome. I'm wondering if the ciliates are able to pull this off because they have that subcortical alveolar structure. So it allows them to mess around with their membrane more.
Yeah, I think that's clearly, and this is one of the key reasons for ciliate behavior and morphology changes to begin with. That architecture that I showed you about, the cortical architecture of microtubule anchored on membranes, that's a very classic ciliate architecture. And without that architecture, none of this behavior is possible. And not just in this system, we've actually done work with several other ciliates where we have shown that this architecture is critical. Now, one of the beauty of this is the fact that you have a large range of geometries. And of course, one of the things in terms of being able to build the class of, there are two really clear directions for us. One is, of course, being able to understand the subtle aspects of geometry. And much of this is just differential geometry that leads to these types of behavior. And then the second aspect is how the cell actually builds it to begin with. So there is a new work that we are working on currently in which we allow, we cut the neck of this animal and we watch how it builds itself. And that allows us to truly understand what are the sets of programs associated with self-assembly of these architectures too, which is not trivial. Like it's not, I mean, it's literally developmental biology, but at a single cell stage, because these sets of architectures are so complex and so precise. But the amount of precision that ciliates show in building their cortical cytoskeleton is absolutely incredible.
Absolutely. So the only thought that I had sort of as a counterexample would be a euglenoid that also has the diagonal
but they also have
striations
cortical
and the pellicle, but no
architectures.
subcortical alveoli.
Yes.
Right. So I'm just wondering if there's anything there that
I think we've looked at them too.
Because
And one thing that we showed is that's a shear-induced system, while this is truly origami. And we can actually also see that in a membrane, what are the boundaries between when you primarily get 2D flow versus with all of these microtubules landing on the surface, they actually suppress a significant amount of rearrangement of the membranes to a point where you can think of them as an origami inextensible-like architecture. So it's quite interesting that you mentioned that. And, there's also an interesting mechanobiology debate going on in the community in mammalian cells of how much information is actually propagated through the membrane when you pull a membrane tether. And of course, that question is moot unless you pin down and say what the cortical cytoskeleton is doing. If you have actin right under there, it's going to propagate information very differently versus if you don't. So in a bleb, if you don't have that actin, it's not going to propagate. So it's actually quite subtle. We use the word membrane often in the community, but membrane makes no sense in a cellular context unless you're able to describe everything that's right under it and as a unit. And so when we are trying to say replicate some of these functions, we should be very careful about building the functional unit of a membrane or an envelope. Because a membrane by itself is not what even just purely diffusion of molecules to a membrane are so controlled by what would occur in the cortical cytoskeleton. And certain cells are able to completely program it, while other cells keep the cortical cytoskeleton quite stationary.
Right, right. So dinoflagellates might be the other. Anyway, thank you very much, and I'll be in touch.
Yes. Yeah, no, I think it would be fun to bump into each other actually in person. Any other questions?
Maybe I'll just say one fun thing.
Yes.
You know that Richard Feynman actually worked on this problem of the garden hose, and especially the reversal, like if you
That's
suck
correct.
liquid in, and then the stability is, altogether different again.
Yes.
And they did it for rigid, pipes, and it's already very hard to solve.
Yes.
So if you have flexibility, I don't think anyone's looked at that.
Yeah, no, I read that old paper, and I think one of the things that there was actually a big controversy around this because
Exactly. Yeah.
oil rigs, you need this because when they get a boat and they get these massive pipes down, and rockets needed it because rocket is literally a rigid rod with a follower force, and any modes of vibration in that system will lead to instabilities. So there's a little bit of rocket science in today's talk, primarily because of that. And all of those were done primarily because none of the trajectories of many of these projectiles could be predicted because it's a chaotic system by definition. Any modes of vibration, and then came this idea that we can decouple. So now a lot of thrusters are actually decoupled from the rocket body on passive balancing systems, and all kinds of control is built right there. Yeah, no, I think the Feynman anecdote is it's quite a puzzling system. It's although very simple, but I just love the fact that cells, whenever there is a mathematical oddity, there is a cell out there that's utilized it.
Thanks, Manu. I'll see you later.
Very fun talk. I'll put out a last call for questions. I know we probably all have added to our reading list pretty significantly. And I'll give another thank you again to Manu. We're going to have you back and hopefully cover all those slides that we didn't get to see.
Yeah, maybe one day I'll talk about algorithmic control of mineralization. I think there's clear certain rules there to learn that would be fun to talk about with this community.
That'd be fantastic. So thank you again, and thank you all for joining us today. I'm going to end the call here. Have
Bye,
a great week.
everyone.
Bye.
Have a good week.